friends, welcome back to my channel, and today in this video we are going to study about complementation test. So what do we mean by complementation test? Complementation test is a test which is done to check whether a mutation, deficiency or loss of function is allelic or non-allelic. That means, in other words, we can say that whether the mutation, deficiency or loss of function is because of same gene or different gene. Allelic means same genes because alleles are the two forms of a same gene. So is it allelic, that is same gene, or non-allelic, that is different gene, that we can test using complementation test. Now for example, we have two mutants, mutant 1 and mutant 2. If by crossing both the mutants we are getting mutant in the next generation, that means that such mutation happened in alleles. The mutations are allelic, that is, it happened on the same gene. In this mutant 1 also, gene 1 is mutated. In this mutant also, gene 1 is only mutated. Hence, after crossing over, because from both the parents, the mutated gene is only passed on to the next generation. Therefore, in the next generation also, as a phenotype, mutant only developed, because from both the parents the mutated alleles are transferred to the next generation. Therefore, if in the next generation we see mutant as a result, that means that is allelic, that is same mutation, same locus, same gene. In other words, we say no complementation. Now if two mutants are crossed, in this mutant gene 1 is mutated, in this mutant gene 2 is mutated. So 1 copy, because from both the parents we get two 1 copies. So in this wild type, in next generation we will get 1 from M1 also and 1 from M2 also. Now suppose this M1 is mutated, but from M2 this gene is normal. Similarly, in M2, 2 is mutated. So here also 1 copy from one parent, another copy from another parent. So one gene is normal. So for gene 2 also one gene is normal. For gene 1 also one gene is normal. Hence, because of this normal allele of both the genes, the next generation become wild type. Whereas in case of allelic mutations, what happen is gene 1 is also mutated from one parent, gene 1 from another parent is also mutated. Now, in the next generation, these two will combine. So, definitely we will get only the mutants. So, when we get mutant in the next generation, it means the interaction or the mutation is allelic. When we get wild type, that means it is a non-allelic. So, here we call it as a complementation. Both complemented each other. The loss of function in one parent is complemented by normal gene in the another parent. So this is known as complementation test. So with the help of this test, we can know that whether the mutation happened in same gene or it is in different gene. Now, for example, this is one pathway. Ornithine is getting converted into citrulline. Citrulline is getting converted into arginine. To convert ornithine into citrulline, some gene A is responsible, which produce enzyme E1 that helps to convert ornithine into citrulline. And similarly, for the conversion of citrulline to arginine, some gene B is involved, which produces enzyme E2, which converts citrulline into arginine. Now, 4 mutants of Neurospora, mutant 1, 2, 3 and 4. Mutant 1, mutant 2, mutant 3 and mutant 4, in all the mutants, arginine is not produced. Why? Because in mutant 1, gene A is mutated. In mutant 2, gene B is mutated. In mutant 3, again gene B is mutated. In mutant 4, gene A is mutated. So, all the 4 mutants, they are unable to produce arginine because of either mutation in the gene A or mutation in the gene B, because both the genes are needed to form arginine, because arginine is a two-step pathway. Ornithine should first get converted into citrulline, then citrulline should get converted into arginine. So either gene A is mutated or gene B is mutated. In both the condition, arginine production will not happen. So in these 4 mutants of Neurospora, either gene A is mutated or gene B is mutated, and because of which they are unable to produce arginine. Now, on the basis of this data, if you ask to find allelic mutants and non-allelic mutants, how you can find that? Allelic mutants, see here M1. In M1, A minus B plus. Again in M4, it is A minus B plus. So that means same gene is mutated. So M1 and M4 are allelic. Similarly, M2 and M3 are allelic. Then what are non-allelic mutants? M1 and M2. Because in M1, A is mutated, in M2, B is mutated. Similarly, M1 and M3, then M2 and M4. So like this, we will get multiple non-allelic mutants also. So with this data, we can easily find out, we can easily see what are allelic mutants and what are non-allelic mutants. Sometimes we will have a table like this. On the basis of the table, now we have to find the allelic and non-allelic mutants. How we can find that? Plus symbolizes complementation and red circles symbolizes no complementation. Complementation means we will get wild type. That means mutation in separate genes. That means these are non-allelic. So wherever we will see this plus, those are all non-allelic. Clear? So where you can see plus here. This is a connection between M1 and M2. So M1 and M2 are non-allelic mutants. Here also, with this information, here also we obtain the result M1 and M2 are non-allelic. From the table also, we are getting M1 and M2 are non-allelic. Then here plus is there. So M3 and M1 non-allelic. Then here again M2 and M1. So the same. Here M2 and M4. This is M4 and here M2. So M2 and M4 non-allelic. Then here M3 and M1, which we have already written. Then here M3 and M4. Then here M4 and M2, which we have already written. And here M3 and M4 also we have already written. So from the table, from the tabular information also, we can able to find allelic and non-allelic mutants. And also when we are given like this, that is already it is written A plus, B minus, B plus, A minus. Like this, when it is written, it is easy for us to find the allelic and non-allelic mutants. But from the table also, we can easily find allelic and non-allelic mutants. Now this is a homework for you. Here to form histidine, first, the conversion needed is A to B, then B to C, and then C will convert into histidine. For the conversion of A to B, we need 1. From B to C, we need enzyme 2. For C to histidine, we need enzyme 3. So wild type means when all the 3 enzymes are present. So these are the 6 mutants. In mutant 1, 1 is mutated. In mutant 2, 2 is mutated. In mutant 3, 3 is mutated. In mutant 4, 2 is mutated. In mutant 5, 3 is mutated. In mutant 6, 1 is mutated. So on the basis of this information, you have to tell me allelic and non-allelic mutants. And similarly, you have to tell me the allelic and non-allelic mutants on the basis of this table as well. Though the result will come the same, but I will ask you not to copy the result, but try to solve separately from table also, so that you can learn how to get allelic and non-allelic mutants from the table also. So do let me know in the comment section if you found the video informative. Please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.